My name is Ramsey Zaitoun, and I'm with Twist, obviously in the R&D department. And last year we came here at AGBT and we released our target enrichment system. We released our exome, and we were, pretty excited, pretty proud of what we developed. Was a pretty highly efficient target enrichment system. Great uniformity, great customizability to make probes and panels. And we came here, had lofty claims, and we had about a year to get tested by everyone, and we got tested pretty hard. And I think we passed the test. We're here another year for that. So I just want to give you a little bit of an idea of what we've done from an R&D perspective to make a good target enrichment system. So basically we just have to say what our mission is, what we're trying to accomplish. And really it's just with target enrichment, what we're trying to do is, sequence the most and get the most information we can with the least amount of sequencing and the least amount of effort possible. That's simple. And to get that, we have two ideas we can push. We can optimize the sequencing efficiency, making sure that we use less sequencing to get the same amount of information, preventing things like off-target, bias, and all that. That all plays into account. And then there's sequencing effort. We want to make it so you have to spend less time and less energy to get the information that you need. And I'll go into those two and how we've taken data-driven approaches to do that. So, sequencing efficiency. So this is basically an idea of a capture profile. You can have on target, trying to capture targets. You can have off target. You can have overrepresentation, underrepresentation. All those come together to represent one thing. We just want to have our targets covered to some minimum depth. I say here 30x coverage, but you can play around with that depending on what you need. But all it comes down to is we want all of our targets covered to some minimum depth with as little sequencing as possible. And the approach we take is this engineering approach of design, build, test, learn. Now, basically what you do is you design a panel of probes, build it, test it, learn from it, iterate. So this iterative approach, it's powerful when you can iterate more. So that's limited by, for instance, the build, when you may need to, the speed with which you can run this process is limited by build. And then, of course, the information that we gain through iterations needs to be concise and reproducible. So that, of course, is limited by build and test. So if it's not reproducible, you can't learn through iterations, right? So first, obviously, this is one of the most important is build, and that's actually making the probes, and that's something we do really well. Else we wouldn't be here. So basically, we write probes on Twist silicon DNA synthesis platform, where we write oligos. And this platform makes oligos with great representation, great quality, and it does it well. And you can make 1 million oligos at one time with little effort. For scale, our exome is 350,000 probes. So we can write an exome no problem. And that's fast turnaround time. We're talking 3 weeks, right? And we have a scale-up process that allows us to control the bias, control the content in the population to get really good quality. So this is a reproducibility graph of 2 lots of the same panel that we synthesized 1 month apart. Didn't tell anyone I was doing this, so to really represent the process, all they heard was an R&D lot. So maybe came out worse. So our first is lot 1 replicate versus lot 2 replicate. Each dot represents the representation of a probe in that panel. And we use NGSQC to do that. We do NGSQC with every panel that we make. And we get really great representation there. So I've shown you the build. We can make these panels really quickly, and we get great lot-to-lot reproducibility. What about test? Well, with test, that's just taking these same panels and actually running them and seeing the quality and the results that you get between runs. So of course, these are 2 panels, again, made 1 month apart, same panel as I showed earlier. And this is the capture for each probe. Each dot represents the capture profile of a probe. And you can see that this the scale here is shrunk. It's smaller because it's more of a uniform capture. And you see great overall correlation between 2 runs from 2 lots. So we have build and test down. We can do it really well. We can make an exome. We can iterate on that, and we have done that. So all that infrastructure that we have here is going and leading into our design and learn steps. So we've done a lot of learning and designing based on that, and I'll give you a flavor of 3 of the things we've done just so you get an idea of how we think about this problem. First is uniformity. Our goal is to distribute these overrepresented reads to lower represented reads, so you need less sequencing to get the same amount of information. And you know, this plays into our design algorithms, but I want to just show an example of how we use this for a customer that was doing a little non-standard of an application that got a GC bias in their profile. So this graph here is probe GC versus the capture rate of each probe in the panel. Initially, they saw a GC bias because they were doing something a little different, where high GC sequences were overrepresented. And they said, hey, you know, we've heard you guys can do this, you know, design, build, test, learn. Can you take some of our data and make a reiteration and balance that out? And we've done it before. We do it with we've done it with to get to where we are in terms of the panels we make and make sure they're good on a first pass. And so we ran it through our algorithm, and we got the great uniform capture, as expected, no GC bias. This is iteration 1, iteration 2 on a fast turnaround platform. It's pretty good and expected. The next question is off target. How do we deal with off target? How do we handle that? How do we take these reads and distribute them to these places so you need less sequencing? Usually the strategy done for that is just to remove bad probes, right? You look at the repeat masker or something and say, all right, this is on a repetitive element, let's get rid of that probe, but that doesn't always mean that you get rid of the off target. Some probes are on repetitive regions that don't actually cause off target. So you're basically just taking a guess and removing content that shouldn't be removed. So those false positives are not a good thing. We don't want that. So we said, all right, let's make sure that every time we identify a bad probe, let's make sure we know what it is and how bad it is. So we took a lot of time building models in terms of and measuring a lot of data to actually be able to predict what a bad probe is, what a good probe is, and the severity of each bad and good probe. So we reduce false positives. We don't just pull stuff out and say, hey, you know, like at a guess, because we don't want to do that for your content. So initially, this was a really bad panel we made. We wanted to, you know, just hit bad stuff. Initially, it was 40% off target. And so we identified, okay, what are the bad probes, the worst ones, the worst offenders? 0.4% of the probes, we removed them, it dropped down to 30% off target. Okay. We have our next bin, you know, 1.7% of the probes total, including that 0.4, drop that under 20%, and so on and so forth. So we have control. We know which groups of probes are bad. We have very good control. And you can imagine, once you know which probes are bad and where things are bad, you can kind of alter your design to work around that and do some pretty interesting application work, right? And last, and this is pretty exciting stuff that's been coming out lately, is just working with applications. So, for instance, all our targets are going to be different. Let's say you're looking at fusions. That's going to be a different application. Viral samples, there's going to be a lot of diversity in there. Epigenetics and bisulfite sequencing, you're going to have a lot of diversity in your sample. So how do you deal with all these different targets? You can't just have probes that do the same thing for everything, right? So we've spent a lot of time and actually looking at how diversity affects probe capture. And since we can make probes really easily and reproducibly, we just synthesized two 30,000 probe panels. That's about a tenth of the exome, a control panel with no mutations in it, and a variant panel that contained design mutations, either SNPs or single mutations randomly distributed throughout the probe, or continuous mutations throughout in the probe, and measured the efficiency of capture between this probe and that probe in 2 different runs. And there's lots of data, lots of hypotheses built into this, including like GC and all that stuff you can imagine. But just some representative data. This is the efficiency before and after. You expect this to be 1 if the probe captures the same, if it's a variant or not. For 3, 5, 10, and 15 mismatches, and at 15 mismatches, you start to actually lose coverage for randomly distributed mutations in our probes. But they capture still pretty well at 10 mismatches, and this is something that we've been using for making more selective captures for different applications. So we can handle applications pretty well by implementing these types of ideas. So, I just gave you a flavor. There's a lot more, and obviously I couldn't share everything here. But we take all that, and now we can make panels on a first pass using all our learning to make good panels. And here's an example of 7 panels that we made. This is the first shot at each panel using our design algorithms. Vary in size from under 20 kb to 13 megabase, no problem to make. And again, remember what I was saying, is 30x coverage is the most important metric. It's the culmination of everything, of uniformity and off-target and everything. And all of these panels get around or above 95% 30x coverage. At 150x sequencing, and also we have to do map quality filtering, very important. But I'm very happy with this, and if this was down here, I probably would have been happy too, but I'm even happier. It's a good thing. Okay, so that's our efficiency. Done a good job with that. I hope you're slightly convinced of that. So let's talk about effort. How do we make it easier to do experiments? So with effort, we think about this three ways. One, we want to make it so you can utilize your sequencer capacity more, so you can put more sequences on a lane of like a NovaSeq or something, right? So you don't there's no barcode clashing or anything. We want you to be able to multiplex your captures, right? And we also want to have fast turnaround time. So I'll talk about those really quickly. For putting more samples on a sequencer, we have a really excellent UDI system that allows you to have indices up to whatever plex we decide to stop at. So it's a pretty good system, and it allows you to just put a lot of samples on a sequencer. So when comparing the UDI system to a standard Y adapter, you see there's a lot of this unwanted product, right? That's not good. You don't want that going into your capture. You don't want that going onto your sequencer, right? With these UDI adapters, they're so efficient that you don't get that unwanted product. And you also get efficient amplification and production of your material, which means less cycles, less bias, cheaper, and less time. So that's one, with making adapters to allow you to utilize the sequencer. But what about multiplexing? You need blockers. Blockers are something that will prevent daisy chaining between the P5, P7 primers or whatever system you're working with. And we've developed a really solid universal blocker system that handles that quite well. In some of the importance of the blockers is that they have to be compatible with your barcodes and indices that you're using, the ones I was talking about earlier. And to utilize sequencer capacity, you need essentially to put as many samples on, right? So we're going to get to a point where people want longer barcodes to get more sequences on, or more samples onto a sequencing lane, right? So you may want to go beyond 8, 10 base pair barcodes. You may want to go 12, 14 base pair barcodes, just so, to really utilize the sequencing the sequencing capacity, and potentially do 10,000 samples on a NovaSeq lane, if you're feeling bold. So right now, our blockers, they work well. This graph here is fraction on target of these blockers with and without in a sample, so you can see their effect on target. If they're not in a sample, it's very low on target. And if they are in the sample, they work well. They do their thing. And the most important point is they're future-proof. They work for 12 and 14 base pair barcodes. So as we expand the portfolio and as technology progresses, these things are going to persist, and they're going to work with the fact that if you need 1,000 samples on a sequencing lane, it'll work. So that's pretty exciting. And you can combine these ideas, this foundation of adapters, blockers, and probes to do multiplexing, right? And this example here is, again, 30x coverage of 3 panels, either 0.8 kb or 800 kb, 3.3 megabases, or exome at 33.1 megabases. And this is the multiplexing performance of 1, 8, and 16 plex. Works well. Everything works together very nicely, and it's, I like using it. So finally, last thing I wanted to say is talk about the speed when we're talking about effort. So we came out with a 16-hour Hyb, an overnight Hyb. So you set it up, go home, come back the next day and finish it. But we wanted to do a single-day workflow, and we wanted to do it well and do it as fast as we can. So we decided to shrink this 16-hour Hyb into about 15 minutes, or whatever time you need. We wanted to give you the flexibility there. So we developed a buffer system that would allow you to do that. And so our standard our 16-hour Hyb now is between 15 minutes and 4 hours. You can choose whatever you want. there's a little, you can imagine a small performance drop. It's still quite good at 15 minutes, but I mean, you could also do 4 hours too. So it's a good system. It works really well, and yeah. So just those two ideas, efficiency, effort, we've been doing our best to address, and I think we've done that, and I hope I've shown you that. And also make sure to see these posters, 512 and 816, that have this information expanded quite a bit. And yeah, just wanted to acknowledge the technical team that's been working pretty hard on this the past year. So if you see these faces around, yeah, make sure to, and you want to talk about some of the technology, and of course anyone in this room can probably, or with a Twist shirt can probably talk about it, but yeah, make sure to say hello. Thanks. Yeah. Bye.